I'm Bob Deans, and I'm CSO at Synthego. Synthego is a genome engineering company that's currently located in Redwood City, and I wanted to give you a backdrop on both the founding and the principles of the company and some of the ways that we're really pressing to make genome engineering a clinical reality. And so we're really strongly convicted to bringing these therapies forward and live by the word accessibility. And so in part, we do that to drive and see the opportunity to drive cost of goods and bioprocesses and clinical manufacturing down to be able to enable these therapies to come forward. But there's also another element, and that element is pushing the data that's being collected in clinical genetic studies, genetic association studies, but the wealth of information that we can glean from a high throughput engineered disease modeling approach. So these are the principles that I wanted to walk through as we go through the story of Synthego today. And so Synthego began as a company focused on providing research guides for gene editing, primarily to the academic community. And it's grown up tremendously. So you're getting a picture now in the upper right there of about 350 plus employees that are both distributed but also work in our centers of excellence for guide production and manufacturing up in the Redwood City area. One of the things we love, we have an engineering background, we love data, and people organize and collate our data so we always learn and improve. So one thing that we're proud of for the research community are the total number of edits we make. I mean, this is something like counting your baseball cards when you were a kid, but we're addicted to it. So we've also used that data to become very proficient in the types of cells that we edit and the editing efficiency that can be accomplished. And so, although that began in the research community to provide people with accuracy and integrity in their data, it's really clear that those principles are central to be able to produce a GMP gene editing product and open the door to translation. So one of the reasons I was so attracted to Synthego was a conversation I had with Paul Dabrowski, the CEO at the time. And Paul actually did a lot of his training and then entrepreneurship through SpaceX. And so in my past, I've had some background with some high-tech Cambridge groups, some powerful VC firms there, and I realized all of a sudden, here's a chance to see SpaceX meet Moderna. I mean, the power of bringing engineering technology into the life sciences is incredibly powerful. And so I want to show you a few examples of how we're building it and how we're applying that. And so what we currently do is have two strong core platforms within Synthego, one focused on the RNA guide technology, and the other focused on how we effectively, efficiently edit cells for the primary cell lines. And those are really the Eclipse and the Halo platforms there. And if you see where they're characterized, it's really what we put out currently as our commercial offerings. It's really throughput and high quality products in that realm. And so those platforms I'll describe in more detail, but they're based on technologies that we've stacked up that enable them. And stacking technologies and growth is a constant process. We work constantly with partners in developing high efficiency editing reagents, and as a consequence, we learn and know the translational space and path forward. So what you really see us doing is building up platforms that further develop out and bring more power to our discovery platforms, and you're also seeing us apply many of our engineering and robotic automation tools towards the process of cell engineering, cell manufacturing. And so this is really, on the left, the technologies that we've amalgamated up into either the Halo and Eclipse platform. So just short story, Halo equals RNA, Eclipse equals cells as a way to track the basic tenets of those two platforms. But it's really about understanding how these engineering-based technologies can interface with many of the life sciences tools and really help us engineer down the complexity of the problems that we're trying to solve, really reduce the dimensionality of some of the biology questions that we're trying to solve. And so for the first platform, the Halo platform, it's really a combination of these engineering principles and AI ML learning combined with chemistry. So it's not just commercial chemistry to make oligonucleotides, it's chemistry so we can understand modifications to these guides that give us power in controlling on-off target editing or controlling some of the regulatory requirements that we're hearing as we go through the FDA. And so what we have with this platform are a series of offerings that people can take advantage of. And what we're doing, and this will be present on another slide, is really mapping out the stages in moving from discovery to clinic and then understanding where in a genome engineering context help is needed, products are used, and where processes need to be developed. And so you can see here that it's really a stream of thinking around high efficiency guides that are then rolling into GMP processes. So we realized, following the value chain, the tremendous value of GMP guides as the genome engineering space is opening up. And so around that platform, we have some innovation and technology that we think is highly advantageous in the safety area. One of those is from a publication we had about a year ago, and it's where we described modified guides that could contain moieties that were light sensitive, and those moieties would let us turn a guide on or turn a guide off. So we can introduce an RNP complex into a cell and allow it to act and then shut it off completely. Or we could put it in and wait and decide when to turn it on. Our thinking is, and our knowledge is, that the on-target hits that you get in an editing process occur rapidly and quickly. And many of the off-target events that you're chasing come from residual guide, enzyme, or complex that dissipate over time and bring up the off-target consequences. And so we've got a number of ways that we manage the benefit of on but very low off-target analysis by incorporating chemistries into our process. I think one of the things that we're seeing as feedback from FDA communications is that the FDA is very concerned about residual guide or residual protein. And so you can imagine that we're building towards a situation where you can have a complex guide that's activated by light, and because of the light control we have, we can do multiple edits simultaneously and organize the time course at which they appear. So I'm going to shift over to the Eclipse platform. This is really the platform where we're putting our automation robotic skills to play in highly efficient cell editing and then providing out edited cell products. And so those are sometimes immortalized cell lines that are used in drug discovery for target validation. Other times we're doing very similar things with primary cell populations, macrophages, T cells. We have a very strong program in iPSC editing and development, and we're really pleased with how the framework of this platform is building us translationally towards support of clinical GMP manufacturing. And so again, this is a framework of our discovery to clinic sort of segments or tracks, and you're seeing the types of offerings that we have in terms of generating knockout libraries for target validation or for screening, but then also moving forward towards robotic and automated control of the cell editing and GMP process. And so one example that we've had that struck me, this actually came out when I first joined the company about a year and a half ago, but it was work that was done in response to COVID, big surprise. And so we had a relationship with Nevin Krogan at UCSF, and Nevin has been spending quite a bit of time studying how viral proteins interact with human host proteins during viral infection. And so his way of thinking is that if you can identify those necessary contact points in the human cell, you can design drugs against the human rather than the viral protein and likely have a pan benefit. But we basically just jumped on board because it was exciting to do and we wanted to contribute. And so what we did in a 3-week period, we ordered and synthesized over 1,000 guides. We made 300 knockout libraries in 3 cell lines that are COVID susceptible for infectivity. Our knockout rates are 85 to 90%. We did that with one person and shipped those cells out at the end of 2 weeks, and in 3 weeks had hits where we were dropping viral infectivity by a log or a log and a half. And so 4 of those hits turned into repurposed drugs, and one of those is out now in a registrational study. But it's like you start to realize what you can do on a discovery project, both in terms of the dimensions you can probe, but the throughput you can get to meet a need like this. It's just really tremendously rewarding. So here is another example of cell editing work that we've done for the purpose of discovery in the CNS space and using iPSC as a developmentally appropriate cell line for that. And this is work that we did under contract with the NIH. We basically took a panel of over 50 genes, made the numbers that you're seeing here of independent clones, but we made those in both a heterozygotic plus-minus state and in a double knockout state, obviously compared to the double wild type as a control. But we did all that, shipped the cells out in a period of about 4 months. It's just a tremendous way for us to get throughput on embedding diversity in a cell line like iPSC, which has such powerful discovery benefit. So what I want to do now is spend about 4 or 5 slides talking about more specifically the GMP guide process that we have and how we're rolling that forward clinically. So real proud of our current facility and our plans building out a second facility in our center of excellence in the Redwood City area. And so we've had great success so far with both our inspections and our regulatory presentations. We're ISO certified. All of the clients we've worked with who have had GMP guides have made it through the 30-day review period without clinical hold. We understand what the FDA is asking for in terms of forward build for critical analytics that are needed for moving gene engineering into the guide space, and we love working with partners collaboratively to hit those kind of endpoints. And so we really see the field growing strongly. And one of the things that should be obvious is that if you partner with somebody like us, or if we partner with you at the beginning for your first guide to go into patient 1, it's really a commitment to go all the way down the road, through the development process, which we all know scales completely. And so we're very much in a position to be able to line up our calendar and our ability to manufacture along the lines of the development needs that companies come in and approach us for. So it's like an open space now without closed calendar doors where we can meet the needs that go forward here. So it's also important not just in terms of time and getting that lineup to be able to advance and scale, it's also important in terms of some of the offerings we have. So this table is showing that we can provide a research guide, we can provide a GMP guide, but we also do manufacturing under slightly reduced air control systems and slightly reduced quality reporting systems, but make something we're calling GMP Light. And it's perfect to use in early development stages where you need to be working with the same machinery and processes making your final guide, but you really want to keep your costs down in terms of avoiding use of the GMP material until it's absolutely necessary in engineering runs. So real happy to have you talk to people that can give you more information about this, or I'm happy to talk with you about how you can learn more through Synthego. And so what we're really telling you is that we've got a good eye, and we have a lot of people within the company that have translational experience. We understand what the problems are that we continue to build for. Some of those are still low cost of goods. It's patient time in terms of window for treatment, and it's really taking this whole approach of engineering the biology all the way through clinical GMP manufacturing. And so what I've talked to you about with the Halo platform and the Eclipse platform really are setting the stage for you to see between the lines that we've really effectively put together the elements of an operational genome engineering company. So we can go from discovery all through target validation, all the way through clinical product and material, and you're going to see how exciting it is when we approach the clinical manufacturing side as well. It's just an excellent opportunity for us in terms of partnering and continuing to build on the translational ladder. So this is my final slide, and it's really just introducing other members of the team. But I also want to point out a name that you should remember, and it's John Tan, our president and COO. So John spent significant time at Thermo Fisher and significant time at Amazon. So John's the guy that gets you the packages there in 2 days. So again, going back to this, like where's technology taking us all, we're now starting to look at engineering applied to biology, and we're starting to look at operational thinking in order to be able to build and provide these materials to the clients in the translational space. So with 22 seconds left, I'll thank you all very, very much and happy to answer questions after the meeting. Thank you.